Okay, so in the first part of this lecture set about allelic interactions, we introduced a new mode of inheritance, and we called that new mode of inheritance incomplete dominance, which is the expression when you see the heterozygous condition where it's somewhere in between the two homozygous conditions, right? We use examples of snapdragons with a true breeding white and a true breeding red parents. When you put those together and produce a heterozygote, the heterozygote was pink. We talked about at least one model, and there are many, many versions of how this actually occurs, but we talked about one model of how this might happen, which is this haploinsufficiency idea that you simply, one allele cannot produce enough of the protein to make the full color change or something. So we gave you at least one molecular mechanism for that. Now, what we want to do now is introduce under the same sort of group a second mode of inheritance, and that mode of inheritance is called codominance. Okay, now, what codominance is when both of these alleles that you get are fully and simultaneously expressed. So there's no haploinsufficiency or anything else. You express whatever you got from mom, whatever you got from dad, and they both equally are expressed as an outcome. Now, if you look on the web again or in other textbooks, you may find that they blend together incomplete dominance and codominance. Sometimes they just mention incomplete and include codominance as part of that. Others use codominance and blend in incomplete. I think they're separate enough that it's important that you recognize there is a distinct difference, at least at the phenotypic level, about what's occurring. Because if codominance, let's say the snapdragons were codominant, what you would expect to see there is, in fact, some parts of the flower being bright red and some parts being white. They both ought to show up simultaneously, but they don't. They blend together. So that's the example we're using here is codominance, where we've got full expression of both alleles. A possible example of this might be something like roan cattle. I know those of you in agriculture realize roan is a broad term meaning lots of different things, but we'll use it in this example. What we have here is the idea that we've got sort of a solid colored individual, maybe red, and there's this other sort of true breeding color, which might be white. If we think about those as far as this hair color, if we blend them together, what happens here is we don't get a pink animal, right? We don't get a pink cow. We get a cow that has partially expression of full red and partially expression of pure white. And so you actually truly do have sort of this mixed condition that occurs, right? In some stock varieties, this is called a roan. Now, what's important here is that if you cross then these roan with the roan, just like in the when we were talking about snapdragons, you actually recover some of these pure parental types, right? If you cross a roan times a roan, you're expected to get about 1/4 of your progeny being back red, like in this case the parent here. Okay, you expect about 2/4, 1/2 to be roan, and you expect about 1/4 to be white, like this parent up here. Again, much like and very similar to the incomplete dominance, if I know the color in a system like this, or if I know the phenotype, I know the genotype. So with this roan example, if I see a red cow that comes off of this particular mating, a red calf, I know it's a homozygous for the red allele. If it's white, I know it's homozygous for the white allele. If it's roan, I know it's a heterozygote. Okay, so there's a nice perfect one-to-one correlation between the genotype and the phenotype. So the same signal, same type idea that you got with incomplete dominance. Notice again, I'm using R's and W's here to represent the alleles, a white allele and a red allele, okay? And I think that's appropriate. If that's confusing you, once again, feel free to use big A's and little a's if you want. I don't do that because people tend to associate that little a with a recessive and it just gets stuck in your head. And when you see it on a test or a problem, sometimes you just immediately go to simple Mendelian or recessive when in fact it's not. We're thinking about this. However, on the converse, don't confuse this with the fact that you've got a dihybrid system here. We don't have two loci. We don't have a locus controlling one part of roan and another. We don't have there's no, because you're not going to see a 9:3:1 ratio or anything. You simple Mendelian monohybrid is what is going on here. Okay, just a couple of other little examples here, ones that are important. At least this one is often important in working problems, so I wanted to kind of introduce it. There are a number of blood groups. Blood groups are things where you do blood tests. These come from originally back during the First World War, 100 years ago. If someone was injured in battles, a buddy would come along and they would stick a tube in one arm and a tube in the other arm and transfer blood over. And often that caused the person to, even more complications. The reason being, as we know now, is that there are these specific blood groups. And we're going to talk very soon about ABO blood group, but there's at least 100 known different antigen antibody groups within our human blood systems. One of them is the MN system. And the MN system, you get antigen antibody reactions for M or N, or if you are heterozygote, you get the reactions for both. So if I see someone who's blood type M, I know they're homozygous M. If I see someone who's got blood type N, I know that they're homozygous N. And of course, the hets or MN have that. So again, these are nice, perfect codominant expressions. Both the antigen antibody reaction for M and N occur in a codominant setting because both alleles are producing the products, the protein products. Again, genotypes are going to equal the phenotypes. Here's what we get. Just one other place, just because we use it in the lab and you see it in other places, we're going to do some things called microsatellites. They're just pieces of DNA. They don't really have any, they don't produce protein. We're not seeing phenotype. We're going right to the DNA and using that. But that turns out to be a nice codominance pattern, which makes it very easy to code. If we see some individual with two bands, that means they're a heterozygote. If we see an individual's sample with one band, like these two, then they're homozygous. And microsatellites are very nice because we see a lot of heterozygotes there. We call it heterozygosity. So it helps us determine what's going on. And a lot of those CSI programs and things really, while they might be doing it on a fancy computer program, they're actually generating these types of patterns and looking at them. And they're very simple. Okay, that's a heterozygote for this allele up here. We might call it 100 and maybe 90. This is a heterozygote for the 90 allele, but maybe for the 40 allele down here. And this is a het for 40 and 60 or something, whatever numbers we want to give to them, right? Okay, so like we did in pretty much all of these examples with transmission genetics, so when we introduce a mode of inheritance or a new idea, we like to work a problem, kind of put it in some perspective. So let's work a couple of problems here. They're pretty straightforward. Right? So here's a problem thrown out at you. In chickens, the locus governing flower color is codominance. Present absence of feathers on the legs is simple Mendelian. Okay, so think about what we've seen here. We've got one trait, which is the color of the feathers. All right, and then we've got a separate trait, which at another locus, which is controlling whether or not you have feathers on the legs or not. So this is a dihybrid problem, right? And it's a dihybrid problem with a kind of important distinction because we have codominance in one trait, right? This color idea, and we're going to have a simple Mendelian outcome in the second trait. So now we don't have just simple dominant recessive at one locus and simple dominant recessive at another. We've got two modes of inheritance playing a role here. Okay. Now what's important is the Mendel's Big Six hasn't failed you. Okay. You're going to get for each of these crosses, genotypically exactly what you expect from Mendel's Big Six. I always have mine in my hand or near my desk. You're going to get this outcome over here. If you cross one of these, you're going to get this, okay? Now what we're going to have to do is think about, okay, we don't necessarily get that phenotype that we expect here. We may have to get another. And then when we go to build the assortment ratios, so we build the big ratio, we expect something else. And I can tell you right away, you're not going to get 9-3-3-1 when you do a codominance with a simple Mendelian. All right, so we would have to have some other words down here to tell you about it, but we'll just sort of make it short. We'll say that there's color, there's two alleles, a black and a white allele. And in these chickens, you get three colors, okay? You get black and you get white. So black is the homozygous for the black allele, white chickens are homozygous for the white allele, and when you get the heterozygotes, you get slate blue. Now you're going, well, wait a minute, why is this codominance and not incomplete or something else? Well, the reason is, and again, this is not a chicken, obviously, it's happens to be a turkey, but it's the easiest picture I could find on commons. This is a blue. It looks blue to our eyes, but there is no blue here, okay? What's happening is that you're getting simultaneous expression of black and white in the feathering, and as light passes through the feathering, we perceive that as a bluish tinge. So these are called slate blue, right? And so, but it's true codominance because you're actually getting expression of the black allele and the white allele, so we see blue. Just like in this parrot, right? It's not codominance here, but there's no green in this parrot. This parrot's not a plant. It doesn't have chlorophyll. It doesn't have algae growing in its feathers or anything else. There's no green pigment laid down here. Okay, there's no copper or anything else. This parrot is not green. Okay, this parrot is reflecting as light goes through the feather system. It's reflecting out to us. It's grabbing other parts of the spectrum, and what's coming out to us predominantly with our vision is green. Okay, in the same way here. Okay, so the fact it's not truly blue and doesn't matter. The other trait, simple Mendelian, is legs, presence or absence. It turns out that we're talking about these feathered legs, which are very prized often for show animals. Simple Mendelian system. Simple Mendelian means dominant recessive, right? It's another term the way we use it. So if you get feathering alleles, you get feathered legs, and the only way you can have no featherings, like maybe this turkey has here, would be the homozygous recessive, and very often that's what we see in stock animals because we don't want all this extra feathers to have to take off mechanically when they clean the chickens up. All right, so we've got that. Now we're going to make some kind of cross, obviously. So the cross that we're going to make says something like this. A black chicken with feathered legs derived from pure breeding stock. Okay, so it's telling me it's got feathered legs, right? It's telling you something about what's going on up here. Okay, telling you this black, so you know what's going on over here. It's crossed with a white chicken, so it's going to have some combination genotype like this with featherless legs, so no feathers is right here. Okay, so what are you going to get from that? In other words, you should automatically be thinking, I'm doing nothing but a diagramming problem, right? I've got it set up here. I've got a classic diagram. And in fact, if we really look at this, what we've got here, we've got a homozygous chicken here that's black, right? It's got feathers, but we're told that it's pure breeding, so that means it's homozygous, okay? We then cross it with a white chicken who's homozygous and has the no feathering over here. So it is homozygous for the alternate allele for feathers. What does that look like? I mean, how many times have you seen that cross? If you've done the Mendelian chapter and you've worked the problems, that's a classic dihybrid parental. Okay, why? Big A, big B, big B. Little a, little b, little b. That's all that is. We're using different letters, but it's still just a classic parental dihybrid cross. And we know from that, because this is a dihybrid homozygote, that we're only going to get one gamete type. You're going to get some black genetic information passed down and some feathering information. From this one, we're going to get some white information, the alternate allele, little a if you want, and some no feathering, the recessive for feathers. Put those two gamete possibilities together, and of course, we're going to get this, right? We're going to get BW, which is blue, and we're going to get F, little f, which is feathered. So all the progeny ought to be blue feathered, okay? Now, if you're ahead of me, you can guess what the next thing is. We're going to cross some of these and make an F2, right? What is A times B gives me C, and usually I have to produce something else from it. Sure enough, that's the next question, right? Several pairs of these F1 progeny offspring are allowed to mate. What are you going to get, okay? So we're crossing a dihybrid het now. You could think of it as big A, big B, little b if you really need to, but hopefully you're with me now. So this is a slate blue feathered heterozygous for both of those times one of its mates that came out, and it is also slate blue and feathered. Okay. Again, Mendel's Big Six has not failed us, right? We're simply doing this cross right here. Big A little a times big A little a. Going to be a 1 to 2 to 1 ratio. Now, because of codominance, right? That's what we have here, codominance. We're going to get direct relationship between the genotypes and the phenotypes for this locus. So we're going to see one black for every 2 blues for every 1 white, right? That's what we expect. It's not the same for the feathers because that's a dominant recessive system. So we're going to still crossing big B, little b times big B, little b if you want, or big A, little a times big A, little a, whatever, however you want to think of it, het times het. But because of dominance, genotypically we get a 1 to 2 to 1. Phenotypically we get a 3 to 1, okay? Now, you can just use the same little rules because we want to know the final assortment ratio. Anytime we diagram and we always figure out the last ratios, whether it's segregation or assortment. These are the segregation ratios, so let's figure out what the assortment is going to be. Just take that big B right there and put it with some of these. 1 times 3 is 3. 1 times 1 is 1, okay? If we come back and we think about these guys in here, With that, we just keep crossing all those possibilities, and finally, the white homozygous white with feathered, homozygous white with not feathered, we end up with a ratio that looks something like this. Okay, it's a 3 to 1 to 6 to 2 to 3 to 1. Okay, no reason to memorize that. You can create it anytime you want to. Right? What is this? It is a final assortment ratio. That's what we're going to call it. It's very important. You need to know what that means. Final assortment ratio. Assortment, 2 or more loci. In this case, one involves codominance, the other involves a dominant system. Okay. Later on, we're going to throw codominance and sex linked and codominance and all kinds of things at you. Doesn't matter. It's this simple. It's never going to get more complicated than this. Okay. If you know what the genotypes give you as far as phenotypes, and you know what genotypes you're making, then you can figure it out. Okay. One very important thing: this is not a 9-3-3-1, right? Even though you have 1 to 2 to 1 and 1 to 2 to 1, it's some other ratio, but it adds up to 16 parts. Just burn that 16 in your brain. Okay, it's very important. I'm gonna come back to it over and over again. When you see 16 parts in a problem, 1/16, 5/16, 6/16, 9/16, whatever, it's a dihybrid het problem. That's what it is. I mean, the way you get 16 parts is to take a dihybrid het times a dihybrid het. Now we can make them with trihybrids and things, but you're not going to see that in our problems. Okay, 16 parts is always going to be big A, little a, big B, little b, big A, little a, big B, big little b, every time. Okay, for us. So just get that, learn it, right? And now it may be a different ratio that adds up to it, but once we know that, we know that the parents had to be dihybrid hets. All right, let's try a very similar, simple problem that might be thrown out at you. Okay, you open a page with tests. Oh, goodness gracious, what in the world is all that? I've got to read all this stuff about polled. I don't even know what that means. And okay, you don't worry about it. Polled condition is dominant, right? So whatever polled is dominant. Turns out polled just means you don't have horns. Okay, and horned is the recessive condition. Okay, so big A is dominant over little a. Coat color is determined by codominance, where you've got red, roan, and white, just like we've had before. Okay, so we're just going to immediately start thinking. All he's going to ask me to do is some kind of cross, right? Some simple diagramming. I mean, he may tell me that I've got a homozygous polled red, and I'm crossing it with a homozygous horned white or something. Okay, but whatever it is, I should be able to figure it out. We're going to save a step. We're going to start you at the F1, okay? So in this case, so we would have produced this maybe from there. So what do these guys look like? Well, they got a big A, which is dominant over little a, so both of them are polled, so they're animals without horns. They are heterozygous for the second locus. We just look up here and figure out what that means. It means that they're roan, right? They're right here. So these are polled roans, both of these, okay? Now, what are they going to make, right? So this is a top-down problem. This is A times B, and it's asking me to tell me what C is. That's all it's doing. How do I do that? Mendel's Big Six, right? This cross, because of dominant recessive, it gives me a 3 to 1. Genotypically, it's a 1 to 2 to 1, but we know because this is dominant here. All right. And then the other one gives me a 1 to 2 to 1. Does that look familiar at all? It's exactly like the problem we just did with the birds. Okay. It's just that these are reversed. All right, no big deal. Okay, we put this 3 with this 1. Okay, this, and then we put this with this and this with this, do all the possibilities. So 3 times 1 is 3, and they're all going to be polled. Everybody in this column is going to be polled because they got a big A, everything that's put up with this. And then the color is going to be determined down here. One's going to be a red, we're going to have some roans, and we're going to have some whites. And they're going to be 3 times 1, 3 times 2, and 3 times 1, right? Then we come back and we grab this recessive over here, which is going to be the horned. So everybody that combines up over here is going to be horns, and we just write them down, right? In this case, we've got a horned red, a horned roan, and a horned white, but the ratios are 1 times 1 is 1, 1 times 2 is 2, whatever. We got the same ratio we had before, right? The final assortment ratio is a 3 to 6 to 3 to 1 to 2 to 1. Now, what would we do this problem? Well, you can make problems up like this, right? Make your own up. And then the next question is, oh, I make this cross and I get, eventually my herd is, I keep crossing these same individuals, and my herd eventually is 100 animals or 60 animals. How many of those are going to be polled roan, right? So 6/16 of whatever number are going to be polled roan, or 1/16 are going to be horned red, or whatever. It's easy just to add numbers to these problems, and that's really all it takes. Okay. The important thing for us is, though, even though now we've piled on two modes of inheritance that are different, okay, we've come up, we still get, in this cross, 16 parts. We still see that the segregation ratios are just following Mendel. And this is why this is such a critical little piece of paper. Okay, this column is always going to be correct. We're not going to lose anything there. To make the assortment ratio, just put two of those together, whatever they are. That's all it takes to work these problems. All right, now we're going to move from that, and we're going to talk about more allelic interactions that are slightly more complicated than this, but really the underneath part is going to be just like this. But the mode of inheritance itself, we'll find out, be just a little bit more complicated.